So welcome back everybody. It's now 3:45 and we are in our final session for the day. It's been a fantastic day so far, and the final session is looking at our future, and to chair this session we're going back to David John. So David, if I can pass back to you.
Yes, hello everybody. Apologies, my light seems to have just gone out, so I look like I'm sat in a cave now, but I'm not. I am in my office. So welcome back to our last session. Thank you all of you for staying with us as we continue to celebrate our 90th. As Maya said, we're now going to be looking at the future applications of the CPR. First up will be Dr. Pierre Hélaouët, who works here with us at the MBA, but he's based in France. He'll be talking about one of our new projects called iCPR. After that, another MBA researcher, Dr. Rowena Stern, will be talking, looking at using molecular techniques with the CPR. Thirdly, we'll have a presentation from Dr. Boris Espinasse, based in Norway, who'll be talking about his novel work on zooplankton isoscapes, linking satellite with in situ data. And finally, we'll have Dr. Julie Robidart from the National Oceanographic Centre in Southampton, and she'll be discussing opportunities and innovation in marine biological surveillance. So as the same as our previous sessions, don't forget you can ask questions in the chat, or at the end you can use the raise your hand function, and we'll pose questions to our speakers afterwards. So we'll get going, and we'll start with Pierre.
Good morning. My name is Pierre Hélaouët, and I'm a senior numerical ecologist at the MBA. Today, I'm going to give you a quick overview on the iCPR project. The aim of the project is to integrate new technologies in the CPR survey. 90 years ago, Sir Alister Rady had a simple idea: to map near-surface plankton in space and time. That simple idea became a fantastic one because he had a clear scientific purpose to understand the changing fortunes of the fisheries, and also a clear vision on how to do it, by using a device to be towed behind a ship. 90 years after, that simple idea became the most extensive biological survey in the world, with 61 years of comparable data, more than 300,000 samples containing information on more than 700 taxa. And at this point, it's very important to remember that the CPR survey is using ships of opportunities, or SOOPs. It means that the CPR survey has created a unique network of shipping companies. And this network is the cornerstone of all CPR activities. The idea behind the iCPR is also very simple: to increase our efficiency at monitoring the world ocean. Of course, we want to guarantee the continuity with the previous 90 years, while getting ready for the next 90 years. So, there is a clear scientific purpose to get more information from the CPR, as well as a clear vision on how to do it, by integrating new technologies within the whole survey. First, we want to get more information from the sea by collecting more data. And for that, we developed, in collaboration with Alex Nemo-Smith from Plymouth University, a new module for instruments. This is our first iCPR, in which we added our first module for instruments. This module is composed by a modular payload bay cage that is fitting within a regular CPR, a power housing, a sensor lugger housing, and also a power generation impeller. And this impeller is a key feature for us because it means that the iCPR is self-powered. The first two pictures show you how the module is fitted in a CPR. On the bottom picture, you can see the new propeller in black. This new propeller means that there is no batteries required within an iCPR, which contribute to keep the cost and the weight down and increasing the autonomy. Also, and it's very important, it means that the iCPR is still SOOP friendly. For instance, we have many rules regulating the usage of batteries on a boat, especially if you are in a CPR hanging on a crane. Also, we don't need any special cable, no specific human intervention, and the main feature is the fact that we have more power available, more power to gather new data, but also to be more flexible. Our new module is the ideal platform to add instruments on a CPR. New instruments to monitor the iCPR behavior. For instance, we have a 3-axis accelerometer, a 3-axis gyroscope, and also a depth sensor. And this allows us to better understand how we sample. Also, we monitor the speed and the direction taken by an iCPR by using a magnetometer. And this allows us to better understand where we sample. Of course, monitoring the iCPR behavior is not enough. We also monitor the iCPR environment. For that, we have sensors measuring temperature, conductance, and also we have fluorimeters. And this allows us to better understand what we sample. We developed the module to be the ideal instrumentation platform by being self-powered and flexible. Perhaps the most exciting part of this side of the project is the integration in the module of an holocam. The holocam contains a camera that can measure not only the light intensity, like a regular camera, but also the phase of the light. This is an example of a raw hologram. This is the same hologram without background, and finally, the same hologram at its optimal focal depth. Because of the post-processing required, using an holocam relies heavily on using artificial intelligence and machine learning. This is why we had an MRS student with the University of Edinburgh, EPCC, and he actually finished last week, so congratulations to him. And his project was to focus on developing an artificial intelligence algorithm that can classify the images coming from the flow cam. We also have a PhD student with Plymouth University that is going to start in September, and he will be focusing on using the images coming from the holocam. We also have other collaboration like with France or Norway. The iCPR project is not only about getting more information from the sea, but also from the lab by integrating new technology to our observations. To achieve this, we started a collaboration with Olympus. The aim for this collaboration is to develop new tools to get more information from the silk. The first thing we want is to get high quality images from the silk. Images from the full sample, or images from a specific individual, or even images from a specific part of a specific individual. We want those images and associated comments and metadata to be added in a new databases of image. This database will be used for training and teaching, but also to feed AI algorithm. And this will allow us to push the next step, have AI on silk. AI for automatic particle delineation, automatic measurements, and ultimately automatic identification. All those elements being stepping stones toward the concept of augmented reality analysis. The third goal of the iCPR project is to get more information from the existing data. And the first step was to enhance our data management to store and connect more information by creating new databases. So we enhanced to integrate new data. For instance, we have now full taxonomic resolution data for every routine taxa in the CPR database. We also have now the main functional traits associated with our taxa list. And finally, to complement the biological data we already have, we are also creating a new database of environmental parameters. We also, of course, have to get ready to ingest our new data coming from the sea, as well as the one coming from the lab. But all these are the very clear goal, to generate new research. As part of this new research, I would like to introduce a new indicator we've been developing. It's a community stability index developed for CPR data, but compatible with virtually any dataset. When extracting raw time series from a specific location, like here in the North Sea, it becomes obvious that it's hard to see any pattern, and this is because the signals are all mixed. However, it is possible to model only the long-term signal of any taxa. Then the signals are much clearer, allowing us to clearly identify patterns such as period of high abundances or low abundances. It is also possible to synthesize the information contained in the whole model planktonic community to obtain the planktonic community trajectory at long term. And this allows us to identify the extreme direction taken by the community. Then, taking those trajectories, it's also possible to further synthesize the information to obtain a simple indicator of stability. Indicator which clearly identify period of instability in our selected areas. So, the indicator will help comparing period of time, but also comparing different areas. Finally, the indicator is an enhancement compared to the one using a period of reference or unable to directly work with missing values. This piece of research is directly connected to our focus on using artificial intelligence and machine learning. Using CPR time series and satellite data, we are developing shallow learning models called NARCs. Those models will allow us to model taxa abundance without CPR observation. We will then be able to combine CPR observation with model data to get an almost real-time community stability index. Getting more from existing CPR data is also about revisiting well-known ecological concepts. For instance, Nicola, our new postdoc, is adding functional traits to our CPR database to explore the structure and functioning of planktonic ecosystem. By focusing on functional groups, Nicola gives us new insights on the role of specific populations in a given ecosystem. In turn, this will give us more information on the potential impact of regime shifts. It will also help us creating new planktonic groups, and finally may lead to the creation of a new generation of indicator. Finally, I would like to mention the work initiated by Lawrence, our new researcher. Lawrence is investigating the time frequency relationship in plankton time series. More specifically, he's using wavelet analysis to explore the structure of time series and their potential synchrony at large spatial scales. Those synchrony can exist in time between taxa, but also in time between areas. Those analyses are providing information on the different dynamics of planktonic populations as well as their link with the physical environment. Finally, those analyses may allow us to train artificial intelligence to automatically identify specific temporal patterns. The iCPR is about integrating new technologies in the existing CPR survey. So I would like to say thank you to all CPR staff members and also thank you to you who listened to me. I leave you with the list of all iCPR team members.
Thank you, Pierre. That's a very good talk there, really sort of showing the sort of new areas that we'll be moving into. And, iCPR really is kind of like the next step in the CPR evolution. So that's excellent. So next up, we will have Rowena.
Hello, everyone. I'm Dr. Rowena Stern. I'm a molecular ecologist at the CPR Survey. My talk is going to be about the possibilities of the CPR Survey using molecular applications and tools. So here we see a picture of various different plankton states. Molecular tools are really essential for discovering the unknown element of plankton. You've heard about all the amazing discoveries that microscopic analysis can uncover over the long term, but molecular tools have really come into their own in the last 10 years, yet they are an essential tool for discovering the unknown elements. If you look at this sort of central plankton picture, first of all, we can identify unknowns. This strange circular object is actually an acantharian cyst, which would be nearly impossible to identify using microscopic methods. We've got a picture of a community, and within that community you can see lots of organisms, but even underneath that there are even more organisms that are unknown. And finally, most importantly, we can identify markers of marine health, and this is really what I'm going to be focusing on today. If we have a look at this picture of lots of bacteria, it leads to a discovery by Luigi Fasuli back in 2012, who looked at pathogenic bacteria and discovered how it's increasing with sea surface temperature. We have really made huge progresses in extracting DNA from CPR samples. In fact, I think we're just over 600 CPR samples that have been extracted for either DNA or RNA. And these range from the Arctic to tropics, and a variety of researchers, not just myself, are involved in this effort. So that's paying tribute to that. These are just some of the papers that have come out from this type of analysis. Very early on, Richard Kirby and Alistair Lindley produced a paper on North Sea echinoderms, identifying them using genetic methods. And Ripley and Declan Schroeder's group moved forward with this and analysed these tiny coccolithophores called Emiliana huxleyi in 2008. These methods really served to bring forward the CPR survey molecular analysis. Most recently, in the Southern Ocean, this paper by Suter et al. Had looked at a whole transect of zooplankton and covered enormous diversity, and really found how effective the CPR survey is in capturing zooplankton. In the North Pacific, a study I recently led along with Stephanie Moore and others in the Northwest Pacific uncovered huge range of diversity of the toxic algae Pseudonychia, and that's really a problem over there. And finally, globally, Luigi Fasuli has analysed samples in the North Atlantic, in the South Atlantic, and all over, and the Pacific, and all over the world to uncover how the pathogenic bacteria Vibrio is behaving in response to climate change. This is a summary of how we extract DNA from CPR samples. CPR samples come in a nice little packet, and they're preserved in a final concentration of 4% formalin. We cut them up. Other people just pick off individuals. There could be a range of methods used here. There's essential washing step, and it's a very elaborate process that takes about a week. And you can see these nice little colour ranges. This is a DNA extraction done all the way across the channel. You can see the different phytoplankton pigments that have come out from that, and they go through a range of chemical extractions to finally extract DNA. And this big circle with different colours is the final DNA compared to a range of known standards that are publicly available, and everything in red type that isn't sort of coloured in isn't coloured is a new type. And using that, we can then see the most similar thing that the DNA is linked to, and we can give it a possible identification. So it's really very powerful tool, because you don't even have to know what the organism looks like to begin with. You can find out at the end. So unfortunately, whilst formalin is brilliant for preserving the morphological features of plankton, it's not so great for DNA and RNA. And this paper sort of summarises the various different types of damage that it can do to to formalin preserved samples. The most important damage is that it can make the DNA smaller. If you see on the right hand side this large piece of DNA, and it gets chopped up if it's not buffered. But luckily the CPR survey was already onto that and was making sure that the CPR samples, the formalin was buffered, and that has maintained its quality, which would not be maintained if they hadn't done that. There's various other different types of damage, but what can happen here, as you can see on the right hand side, the red piece of DNA. If the DNA is chopped up, then what can happen is we can miss our target if we want to amplify a specific region of that DNA, and if it's too small or it might be missed. The other option is that it can the formalin sort of cross links with proteins and other pieces of DNA, and it blocks sort of any enzyme action that we want to do in order to extract a specific part of that DNA for analysis. So the key messages here is for the future to capture DNA as near as possible to the collection time to reduce that level of damage as the damage increases with time. I'd really like to sort of talk a little bit more about studies that have been done on Vibrio, and because it's really been one of the most successful examples of how the CPR survey is useful in measuring the health of the oceans. Vibrio globally is increasing. Luigi Vasuli discovered that in 2016, and this is because it's very much a species that directly is affected by ocean temperature. In the United States, Vibrio infections have also gone up by 85%. There are three main species, Vibrio cholera, Vibrio parahemolyticus, and Vibrio vulnificus, that are especially pathogenic. They cause a range of gastrointestinal diseases and skin lesions. And then most of them are caused by consuming contaminated shellfish. So although, as I mentioned, there's thousands of environmental strains, some contain toxin genes, and these can be transferred between strains. So that creates an ever-changing kind of environment in which a non-toxic strain could suddenly become toxic. But there's been sporadic cases of Vibrio infection, and I've put here the colours: green for Vibrio parahemolyticus, yellow for Vibrio cholera, and the years that it happened. So even though the Pacific Northwest has very cold waters, nevertheless these are occurring, and it's not clear why. If we look at this diagram, we can see that sort of Vibrios are a part of a larger system. Climate change impacts a number of different factors such as temperature and salinity and pH, and they all affect the marine ecosystem at different levels, all the way down to the cell with a cascading effect. And what happens at a sort of a large scale can affect how cells behave and respond to that. So one of the key things is that at temperatures over 15 degrees, Vibrios can double their population, and that expands the number of possibilities for pathogenic strains to emerge. So I'm going to move on to a study that we're currently carrying out. It was started with a rare sort of cholera outbreak in March 2018 in the Pacific Northwest coast of Canada, off Vancouver Island. And people had consumed herring eggs, which are normally safe, but in this case, people got sick. The Centre for Disease Control and various other agencies took some samples of the environment and for sick people, and they discovered that the people were infected by a Vibrio cholera strain. I should point out that I know that cholera has most people think of cholera as this the tropical cholera, which is extremely dangerous and often fatal. This type of strain is a self-limiting strain. It's not the epidemic form, so it's a non-010139 cholera strain. The 010139 is the epidemic variety, but nevertheless it still causes gastrointestinal diseases, but the outcome is better. So I can say that the source of this actual strain is unknown. It could be a spontaneous local species or strain, or it could have moved in from other places in the world. So this project was to uncover the sources and dispersal pathways of this. What we did, and you can see the map here, is we actually managed to extract DNA from, you can see over the star, that is the actual infection site, but from the nearest CPR survey sample transect, you can see the square area there. And we used a range of years, so 2012, which was a cold year, 2015, which is a warm year, and then also 2017 and 2018. What we found was a small amount of Vibrio cholera in April 2017, and then the pink bars show we found a level of Vibrio cholera in 2018 in spring, which was around just a month later after that infection occurred. So Vibrio cholera is still around in the spring waters, even though they're cold. And you can see the pink, one of the pink bars are quite high, and we found quite high levels of one. And we thought that although we found the Vibrio cholera presence, we don't know enough about it to identify it to strain level. So what we had to do is a process called whole genome enrichments. That concentrates the Vibrio DNA. So I'll just show you here, in picture A, you can see here's our mixed plankton sample. We extract the DNA, and in the middle we attach that DNA to Vibrio specific probes. And in the third picture, we start to, the probes extract out just the Vibrio content, and we get an enriched sort of Vibrio enriched sort of DNA sample. And picture B shows an example from another paper from Luigi Fasuli in 2017, who tried this method before. And the dark green bars show the level of enrichment that we got compared to what we would have got if we just used sort of plain DNA or unenriched DNA. And what that means is we can also enrich for the particular markers for toxigenic genes. And that will enable us to identify where the toxic genes have come from and where they spread to. Our next step is now to, you can see this very sick person here, is I link it to the strains that were found in the sick people, and also to some environmental factors. There's a number of things going on around that time. In 2015, there was a bit of a marine heat wave that was sort of offshore. We don't know whether that's affected it, and it might be difficult to tell, but there might be some sort of traces of biological or abiotic factors that may have contributed to these sort of pathogenic strains being there. However, in a recent study by Luigi Fasuli, he towed a CPR sample with Lance Gregory, who was really instrumental in making sure those samples were towed all along Lake Tanganyika in Africa. This shows you the sort of the towing kind of program all along the length of it. And on the right hand side, these were the strains that he managed to extract from the CPR samples. And out of interest, they also towed non-formalin towed samples, but he didn't really find anything from there. As you can see, everything in red is this strain, and The species next to it was Vibrio cholera, but it was likely an environmental strain. Everything on the right-hand side is the red with the red bar are the toxic epidemic Vibrio cholera, and you can see the type that we found here was more sort of environmental and potentially not pathogenic, but it's still a useful example of how we can extract specific genomic information from CPR samples, and we can link them to a wider database and really find out what's going on with pathogens in marine waters. So moving on here, one of our future efforts is to improve DNA extraction methods. We are trialing this method called isotachophoresis with Purigen Biosystems in California. This is a really simple method and quick method to extract DNA, and it uses the electrical charge of DNA as a property to, you can see the top images, it's all the DNA is mixed in the sample, and using a variety of sort of buffers, separation buffers and electrical charge, you can get nice pure nucleic acid. You see the bar below from that, and really sort of unchanged. And this really sets the paradigm for how molecular research can be done on CPR samples. We have a system where we can collect the samples. They're stored and extracted in parallel, almost after collection. DNA is extracted, and in the case maybe of with Vibrio, we prepare libraries of Vibrio enriched species for sequencing, and we can carry out a microbial community analysis where we identify what species and strains they are and link them up with and find out. You can see the map there, where they are and where they come from, and we can sort of link these up with further efforts, for example, in the Centre for Disease Control or the European Centre for Disease Control, or for NOAA for better predictions on the conditions, and to warn shellfish, for early warning systems for shellfish farmers, for example.
Thank you very much, Rowena. That was a fascinating talk. I'm always very impressed when we can say that we can identify 800 different types of plankton using taxonomy. And then I see these molecular results and I just think, I've got a long way to go. Okay, so next up, let me just check, is going to be Dr. Boris Espinasse, developing large-scale zooplankton isoscapes using observational data.
Hi everyone, my name is Boris Espinasse, and I'm a postdoc at the University of Tromsø in northern Norway. And I will present my work, which are about developing large-scale zooplankton isoscapes using observational data. So I will start by a brief introduction on stable isotopes. So stable isotopes are naturally present in the environment. So we have what we call the common isotope, which is found in higher abundance. So in the case of the carbon, it's carbon-12. And we have other isotopes, which are characterized by a different number of neutrons. And if there is one more neutron, then we'll get heavier isotope, for example, the carbon-13, which is a stable isotope. And if we do the ratio between the heavy isotope and the common isotope, and we compare this ratio to a standard ratio, then we get what we call the isotopic ratio, which in this case will be the delta 13C. And in the nature, the autotroph preferentially use the light isotopes against the heavy one. And a result of that is you can see here in this sketch, if we have a mesocombs with a phytoplankton community, and which is limited by nitrate concentration, and you add nitrate in the mesocosm, Then the phytoplankton will use first the light isotope, the common one, and at some point the nitrate will become limiting, so they will have to use as well the heavier isotope, and the signature or the isotopic value, delta 15N of the phytoplankton, will start to increase. So when we are like away from the coast in the open ocean, we can say roughly that delta 15N is correlated with nitrate concentration, or at least it works in an environment where the nitrate is a limiting nutrient. And delta 15C is correlated with CO2 concentration, which is in turn driven by the water temperature. So what I just described takes place in the left part of the slide. It's isotope uptake by the phytoplankton. And what we can observe then is accumulation of heavy isotope along the food web. So it's what we call enrichment between the two trophic levels, and is due to the excretion of light isotope. So for example, here we have zooplankton feeding on phytoplankton, excreting light isotope, and then there is an accumulation of the heavy one. And so an increase of the isotopic ratio. And since we know roughly the enrichment factor between the different trophic level, if we have isotope value for juvenile salmon, for example, and phytoplankton, then we can estimate the trophic level of the juvenile salmon. So in this case, it's trophic level 3. Then the isoscapes. So the isoscapes are the spatial distribution of isotope value. So there is an example here of nitrogen or delta 15N isoscapes. So it was made by sampling the studio area and sorting out the copepod and looking at delta 15N value for this copepod, and then interpolating between the sampling station. So to do the isoscapes, it's needed to collect sample over the studio area, preferentially within a relatively short time period. That the isoscape is consistent, and also to have organisms that belong to the same trophic level. And the last point is to take into account the integration time of isotope value, so the tissue turnover. That can go from day 4 to 12, a few days a week for zooplankton, and it can differ a lot for larger organisms depending on the type of tissue you are using. The feather for the bird, for example, or the muscle or the plasma in blood. It's a lot of different possibilities. So the isoscapes can be used for different purposes. For example, to identify the sources of key elements. So in the case of the delta 15C, it can be used to track coastal water drifting offshore. For the delta 15N, it can be, for example, the type of nitrogen compounds which are used in the system. So the isotrophy, for example, will have a different type of values than nitrate-based system. To define an index for certain productivity, so we have seen that when a system is productive, then the isotopic ratio tend to increase. To provide a stable isotope baseline for trophic structure study. So if you have value for example, a species of fish over your study area, and you want to know at which trophic level they are feeding, then you will need a baseline, and so the isoscape can provide that, and you can subtract the value from the isoscape to your fish value and estimate the trophic level. And the last point is to track fish or the organisms when they move into the ocean. An example would be otoliths, so where you can look at isotopic value for different parts of the otoliths that correspond to a different part of the life cycle of the individual. And you can associate this value with or see where they are distributed into your isoscapes and estimate the animal movement. So a concrete example of application. It's a study that I've done in the Northeast Pacific. So I was working at UBC at that time. And the context of this study was an important decrease in sockeye salmon stocks observed over the last decades, for some of the stock at least. And very little was known about how they are distributed in the open ocean and what kind of trophic environment they experience. Even so, they spend 2 or 3 years in the open ocean. And to get more information about that, we decided to develop isoscapes over 20 years, mainly based on the CPR samples. So we obtained about 200 samples, focusing on large herbivorous copepods, and we analyzed these copepods for delta 13C and delta 15N values. And to produce isoscapes, instead of just simply interpolating between the station, we wanted to add some more statistical power into that by using predictor. So we looked at satellite data and produced some parameters such as SST, chlorophyll A, sea level anomalies for the eddies, the bathymetry. We also investigate the mixed layer depth based on ergo floats and wind data. So we produce isoscape for 20 years. So you can see here example with 3 years for delta 13C in the top and delta 15N in the bottom. You can see that the coastal area were not considered because the dynamics there is much more complex due to terrestrial input and potentially resuspension from the bottom. And also we are very interested in getting some contrast in the open ocean because it was our study area and where the salmon were distributed. There's some pattern that appears which can be due to eddies, for example, which are productive area. We also have influence from the continental shelf, let's say, and sometimes from warmer water input from the south. So we wanted to see if we can use these isoscapes as an index for ocean productivity. And so to do that, we look at the correlation between the isoscape value and the abundance in large copepod. And 2 regions appears, one in the middle of the Gulf of Alaska, where we have a positive correlation, and one along BC coast with a negative correlation. So the positive correlation in the Gulf of Alaska is rather intuitive, because if the system is productive, then delta 15N will be higher, and it will be potentially a more favorable condition for the large copepod to develop, and we'll find more of them. But it didn't work along the BC coast, so this area is pretty dynamic. There's a lot of physical processes there happening. So there's upwelling, but it's also in a transitional area between the north and the south, with a current moving from one year to another. And so it tends to hide the change in isotopic value, which are due to only to productivity. So it shows that it can work. This correlation works within an equation, but this kind of transitional area, not so much. So once we have produced these isoscapes, we need to know where the salmon, different stock of salmon were distributed. And I will just very briefly go through that, but we use correlation between delta 13C and SST to estimate feeding runs of salmon during the last year at sea. So once we know where the salmon are distributed, we can extract isoscapes value, and so it will be zooplankton value, and so it will be our baseline, and compare that to salmon isotopic value, so that we have measured in the scales when the salmon are back to the river system. And if we subtract the plankton value to the salmon value, we obtain the trophic level of the salmon. And we have been able to show that there was some differences between the stocks, and that it could explain the different trends observed with some stock doing better than others. So now what I am doing, so I'm based in Northern Norway. I got a Marie Curie Fellowship to develop isoscapes in the North Atlantic. And I'm still collaborating with MBA to do that. So I access to about 600 CPR samples, so mainly Calanus submergicus. They have been analyzed for delta 13C and delta 15N. And I want to produce isoscapes for 3 different seasons. And I will try to compare different statistical approach to see which one is working the best. So there are 2 applications included in this project. One is about exploring changes in Atlantic puffin winter diet. So the isoscape will provide isotopic baseline, and we measure isotopic value in puffin feather. And using both, we can estimate the trophic level of the puffin and see if there is a changes between years. We also want to estimate molting ground location, so where the puffin are replacing the primary feather because they cannot fly for a few weeks and they are very exposed to environmental conditions, so it's important to know where that happened. And with this information, we hope we can help to get a better understanding of why the breeding success is decreasing in the last years. So maybe link with some carryover effect with a lower fitness due to harsher conditions during winter. The second application is about using pre-isotopic composition, so zooplankton, to understand the summer distribution of Atlantic mackerel. So it was observed in the last year a shift of the population from the, or more like extension actually, from the east part to the west of the North Atlantic. And if we have isoscapes covering like this shift time, we can use the data from the isoscapes to say that maybe there was some changing happening in the west part of the North Atlantic that could explain why the mackerel start to be distributed there. And thanks for listening.
Thank you very much for that, Boris. That's a fascinating talk. So something very, very novel for the CPR. And it's amazing to see that actually taking that new novel technique, you're still able to apply it back to ultimately to fisheries, which is what the CPR survey was actually started for all along. So that's amazing. Okay, we'll move on then to our final talk, and that's Dr. Julie Robidart from NOC in Southampton. So over to you.
Do you see that okay?
Yeah, that's great. Thank you, Julie.
Great. So today I'm going to talk to you about opportunities and innovation in biological surveillance. And I'm going to start with this image on the right, which is by Kelly Lance at Mbari. And it's one of my favorite images because it's got plankton in it, obviously. It's got fish, and it's also got a bunch of different observing tools. And I'm really into technology, so it's a beautiful image to me. I'm the head of ocean technology and engineering at the NOC. And I wanted to kind of start with a look forward, and we're going to the next 10 years. So the CPR is going to have its 100th anniversary, and usually you can say what's going to happen in the next 10 years. If you were to ask me maybe in 2010, I could start thinking around that. But to be honest, I have no idea what's going to happen in the next 10 years in marine biological observing. Everything's escalating at the moment. So there's a lot of investment in data binding and analysis in technologies for marine biological observations. So it's going to be a different world, I think. And partially that's because the Ocean Decade and the Futures of the Seas and Oceans Initiative. So these are basically programs that are trying to coordinate globally to look at different things that have to do with the ocean, but there are several programs that have to do specifically with biology, and I've listed some here that are relevant to the CPR survey. In addition, there's the SDG 14, Life Below Water, and there's also some around the food sources. So there's this interest in fisheries and marine life. So there's a lot of challenges with new initiatives. So when we go out to observe biological phenomena in the oceans, if you've got a new program, you've really got to think about standardization and interoperability. Those are major things that allow you to look at different data from different areas and understand them. Often there's inconsistent metadata, so patchy measurements that are taken in collaboration with the biological observations. At the beginning of these initiatives, there's low time and space resolution. Data access and analysis is usually a problem if they're complex types of data. And then the integration into policy is key, because that, as Abigail pointed out earlier, requires knowledge, not just data, right? So you can't just inform policy based on the data itself. So the CPR survey has this rich history where it's been able to actually collect all sorts of metadata and CTD data and now even fluorometry in collaboration with all the CPR survey transects on the ships of opportunity. You've blown me away with the 700 taxa. I know, David, you said that it doesn't seem like much, but actually I usually do some targeted analysis, and 700 taxa is a lot. That's a lot of biodiversity. And honestly, with our sequence data, sometimes we can't identify them. So these are actually really useful data compared to sequence data sometimes. I looked up at my favorite organism, Trichodesmium, on the top right here, and Trichodesmium, you know, you've got a time series going to 1958, and here's all the North Atlantic transects. It's insane, the temporal and spatial coverage. So you guys have kind of laid the groundwork for a lot of the biological observations, and there's going to be a lot that these new initiatives can learn from the CPR survey. And there's going to be a lot of upscaling. So what I'm calling upscaling is basically changing how we do biological observations by leveraging different data types. So there's different types of biological data, but also satellite drive data. So the CPR survey is, you guys today have shown a bunch of different demonstrations of using satellite data to even measure phytoplankton, but also to look at zooplankton, which is really cool, using proxies from CPR data. Argo float data are really helpful because they tell you where ocean currents are to look at more regional and mesoscale phenomena. But in addition, Argo floats are soon going to be integrated with particle imagers. So we'll have also some plankton data from the Argo floats themselves that we can use for upscaling. And then molecular observations in fixed observatories. So this is the EMOBON, which is a brand new European marine sequencing biodiversity observation network. And the idea here is that these are fixed locations, marine labs that have all sorts of biodiversity data, and now they've been augmented with sequence data through EMOBON, and that's led by the EMBRC, and that's just started in June. So all of these complementary data types are really going to allow us to do novel things with biological observations. So there's this interplay between the environment with the imaging data, and so the context, the environment, as well as the imaging and molecular. So you can pull out from here things like large scale change using satellite data. You can understand the chemical contexts using in situ sensors, biodiversity data, and a historic record using the CPR survey. And if you integrate these with molecular data, you can add on some functional potential and activity, as well as environmental response. So these are DNA, RNA, and protein data that kind of tell you what the organisms are doing and what they're capable of. The Ocean Biomolecular Observatory Network is basically been kicked off as part of one of the Ocean Decade programs, and it's doing this exact thing. So it's trying to enhance fixed observatories with sequence data, so DNA, RNA, and protein data. It's going to be doing training and technology development in order to increase the observatories for biomolecular data. And it's a good time to do this because, as you guys are all well aware, there's a global pandemic. So the pandemic has actually really initiated a lot of capacity building with sequencing of novel strains, but also it's demonstrated how impactful it could be when we work as a global world of scientists to actually look at different strains and how they're moving around the world over time. Data visualization is key, and these data are usually shared in real time as the sequences are generated. So it's really inspirational to see that. But in addition, the timing is great because there's a lot of initiatives to sequence genomes of all of life. And this is based usually on voucher specimens. So these are organisms that are taxonomically identified that we know are important. And then and those genome sequence data actually enable us to do more with the in situ biomolecular data than you would be able to do otherwise. So if you filter down some seawater, we get a lot of data from that, but often we don't know who the data is assigned to. So this basically gives us a better database to identify those sequences. Environmental DNA is basically that. It's filtering down seawater, sequencing DNA, and or doing targeted analysis of DNA in that seawater. So you're filtering seawater because you know the organisms have been around there and they've sloughed off their tissues and things like that. So there's going to be biomolecular fingerprints in this data. So eDNA has been used to measure a threatened species, Chinook salmon, off the Pacific. And compared to nets, it actually is giving pretty good data when you use quantitative PCR. So we're both based on both abundances and biomass of these fish. You could get a decent correlation with quantitative PCR data on just measuring abundances of the DNA. This has been demonstrated using sequencing tools too of the 12S ribosomal RNA of fish in around New Jersey and New York. So in red there, you're looking at the net data, and this is a normal fisheries abundance count, and then you're looking at eDNA eDNA data in green and blue, and both habitat preference and seasonality were correlated in a lot of these data points. And last, Atlantic cod around the Faroe Islands, it's declining fishery, but they were able to see an 80% agreement in detection using the traditional tools for fisheries abundances and qPCR there. And as Rowena demonstrated just a few minutes ago, you can actually do this for harmful organisms too. So I'm going to give you two examples of Alexandrium minutum. So we've been able to detect the DNA of Alexandrium directly from seawater in less than 40 minutes from sampling to detection. And then if you look at template DNA directly, you can actually amplify and detect using this little miniaturized genetic sensor. There's a picture of it in the middle here that we developed in Ocean Technology and Engineering in less than 5 minutes. And that's a bit faster than what our traditional instrument on the bench. So this is a toxigenic organism. And so you could get really quick data from seawater to detect it in the environment. And most recently, these first studies are from Matt Wilson, who's working in Ocean Technology and Engineering, and these last data are from Ahmed Al-Rafi, who's a student with us. So he's been able to develop an assay to detect the gene responsible for domoic acid toxin synthesis. So domoic acid is a neurotoxin, as Rowena pointed out, and this is the abundances of the DAB-A gene, which codes for that toxin biosynthesis. Those correlated with not only the abundance of the toxigenic pseudonychia, but there was a decent correlation also with domoic acid concentrations when you looked at the RNA. So this tells you a bit about activity of those toxigenic organisms. And as part of Tech Oceans, this project that kicked off in the fall of 2020, we're kind of expanding on these molecular tools and the technologies to try to make miniaturized microfluidic genetic sensors. Genetic sensors already exist. So this is an environmental sample processor, and you can't have a talk about technology for biological observations without mentioning it. So it takes a sample, filters seawater, and it can preserve that filtered sample, or it can extract the cells, do a ribosomal RNA fingerprint, so to get a community analysis, or it could do quantitative PCR on the genes. It can also do toxin detection, and there's assays for domoic acid, sandwich hybridization assays, and it sends the data back to you in real time. So this has been demonstrated in Monterey Bay for pseudonychia. So this pseudonychia is a neurotoxin, but it's been linked to mortality in sea lions around Monterey Bay. There was an ESP deployed here at Station E1 and at the Santa Cruz Wharf, and these are concentrations of domoic acid. The larger the circle, the more domoic acid. So you could see changes over time and then across the bay. But there was also a decent relationship between domoic acid here and pseudonychia, but not domoic acid and the abundances of other organisms. So this is work by John Ryan in collaboration with Rafe Cadella and Chris Scholin. The generation 3 ESP is tiny. It's this little thing right here, and it sits in the nose of a long range AUV. This AUV can go thousands of kilometers, and this ESP takes 60 of these cartridges, which is each of these can collect one sample. So it is able to filter again and apply RNA later, so that you can take that sample and do molecular biology with it when you get the device back in the lab. We have a device to do something similar. So this is our biomolecular auto sampler, and this is the benchtop version and the submerged version. This submerged version has been deployed on AutoSub 6000 just last month, and it filters and then samples or preserves a sample for lab analysis again. It's been pressure rated in this pressure pot to 4,500 meters, and it was deployed to 3,000 meters and took samples there and at the oxygen minimum zone on AutoSub as part of iAtlantic last month. These sequence data are pretty useless without some contextual data. And so we've got in Ocean Technology and Engineering, we're always developing these microfluidic lab-on-a-chip sensors. And these are all the different analytes, the different chemistries that they can measure. Most recently, we've been able to measure subnanomolar iron and ammonium. So those are the newest sensors that are going to be rolling out soon. And I just kind of wanted to end to talk about the CPR program in relation to net zero and to the digital ocean. So these are two initiatives that are going to be pretty important in the UK, I think. So net zero oceanographic capability. Usually people are thinking about autonomous vehicles when you think about net zero. So the idea is to decrease our carbon footprint because we are environmentalists. But in reality, there's these really cool initiatives to make these zero emission cargo ships even. This is out of Norway that just was recently left the docks in August. And then we've got Affinity, which is making the Armada fleet meant to be deployed with a bunch of sensors, but they also can have samplers off the back, so they're large enough to carry a CPR. So this is really exciting. And in addition, the digital twin, people usually think of this as an ocean modeling effort, and that's absolutely part of it. But it's also meant to ingest all the complex types of data that we're generating, especially biological data, because they're really difficult to work with, including historic data, so things like your CPR database, and then responding to and reporting those data. So making online tools that are easily digestible by the public. And so this is great, but it's also a little frightening, because if you see a change in biodiversity, like a decline, you might it might be something that has to do with a normal oscillation of oceanography, but the press might run with it and make some scary projections and scare people. Yeah. So basically, I wanted to get back to this idea that it is really helpful to integrate all these types of data, but actually it's not just the biodiversity and taxonomy data that the CPR program provides, but that knowledge that that Abigail mentioned earlier, that is very key for a brand new biology program. So we just I think we need to be careful about what we say with the data and what our timescales are for detecting change. And yeah, with that, I'll thank the funders and OTE. Thank you for your time.
Thank you very much for that, for Julie. That was fascinating, some amazing things there. I'm already thinking about maybe we could put the ESP on the CPR and perhaps part of the Atlantico project, we could have a think about that. Okay, so Maya, I think you hopefully got some questions for our team.
Microphones if you're a speaker for that final session, and then I will go through the questions. There's a few questions that have been answered, but I'm just going to ask them again just for the benefit of others to hear. So Michael, you asked a couple of questions. There is one about providing references for Community Stability Index, which Pierre has answered saying that he's currently writing a paper. So thank you, Pierre. And there was another one from you, Michael, about have you resolved how the CPR silk samples Vibrio cells? Are they attached to the copepods? And Rita Colwell showed years ago that copepods are a natural host for Vibrio. So Rowena, do you want to just explain your answer to Michael there?
Yes, so before I think we used a kind of a more sort of a method that it was a relative quantification method. I think that in this particular project we used a marker that on the Vibrio that directly binds to Vibrio chitin. Sorry, excuse me, the copepod chitin out on its outer surface. So everything that we detect is Vibrio that is bound to copepods.
Great stuff, thank you, Rowena. And David, you had a question in the chat. Did you want to ask that live to the panelists?
Yes, I could do. I just have to read it again what I put. Yes. Yeah. So I was wondering, do you think we'll ever get to the stage where we'll be able to run sort of in situ, either molecular or other techniques, and use them to identify plankton to species level, and then use that through routine monitoring? So you imagine sort of using the CPR network of vessels, so you could cover all the open oceans, but having something so robust that you could actually swap taxonomy and physical samples for some other system.
So who's that question to?
Well, I suppose it's for Julie, yourself, Rowena, Boris.
I think Rowena could talk about the biology and I could talk about the technology maybe.
Yeah, okay. So I think biologically it's perfectly possible.
it would never replace traditional taxonomy, because there's so much more information that you can get from the microscope to do with it, sort of the condition of the cell. Although even then you could make progress, and associated things. But I think already you can detect things. The level for doing it, on a kind of a sort of a monitoring scale. I'll leave Julie to answer that question.
Yeah, so at the moment, really, we need the databases to be a little bit more rich. In the surface ocean, it's not as bad of a problem as the deep ocean, but it is still a problem. But the in situ sequencing, I mean, I did have a slide that I took out that basically there's components that exist. So there's an automated extraction device. There's a library preparation device and a sequencer that MinION have developed. And they're all miniaturized. Their idea is to get them in remote environments and field-based. And so we're closer to it than a lot of people think, I think. But yeah, it is a challenge. It's not going to be something that's going to happen in the next year, I don't think.
Lovely, thank you, Julie. And we've just got a hand up there from Chris Reed. Did you want to switch your microphone on, Chris? Camera as well, if you like, as a panelist. You're still on mute there, Chris. There we go, we can hear you.
Yeah. Hopefully you can still see me. I'm still awake.
Yeah, we can. Well done. Yeah.
It's fantastic to see all the super ideas for technology. The one concern I have about all of these is that it will many of the people who will be funding the CPR program, the base CPR program, will use these ideas as an opportunity to say, well, we can use technology instead of the CPR. Somewhere we've got to get the message over that you need to do the two things hand in hand. And the other aspect, which I was very familiar with when I was helping to run the CPR, that when you are developing technology, you're spending as much money as you are on the CPR program in terms of staff, and at the same time, and that will continue into the future, and the processing of the data takes a lot more as well. So you need to double your, if not treble, the amount of money that you've got to run the CPR program as you've got at the moment. I'm certain that costs will all come down in the future, but they haven't come down in the last 20 to 30 years of technology to get to the fantastic position that we're in at the moment. So I think all of this needs to be taken into consideration and in the future development. And somehow we need to be able to guarantee that the core CPR program that's given us this 90-year time series is maintained using the same methodologies. I don't know how it can be done, but that's really important.
Thank you. I was going to actually say that this is a key thing that Chris has mentioned. As soon as if you exchange technologies, you've essentially lost almost 100 years of data right there from not just the CPR survey, but multiple long-term time series. So there has to be a way of incorporating and doing things in parallel. So I agree, and maybe you're swapping different disciplines as well. There will always be a need for experts of some level.
Right, thank you.
I kind of wanted to add to that really quickly that yeah, I think that this is a huge area of, it's an issue because I'm in a technology group, right, and I'm writing technology proposals, but yeah, I think we recognize it and we know that the importance of the data, and I think that that can't be trivialized. And I think, I mean, Willie and I have written a few proposals now to collect data in tandem, because yeah, like Rowena said, we're not at a point really where we can use these sequencing technologies, at least, to replace the taxonomic data yet, not in these larger time series, I don't think.
Great, thank you very much. Just while we're talking about the future of CPR, can I just remind you, please, in the chat function, we've got a link to our survey, and it would be fantastic. We've got 60 participants at the moment here. It'd be really wonderful if we get as many people as possible complete that survey. It would be a huge help to us. It's very quick, it's very easy, it's an online survey. So if you could do that before you sign off today, we would be very grateful. If we've got 60 responses, we will be dancing around in offices. So if you could do that, then that would be great. Thank you. Willie, you've got your hand up. Would you like to put your camera on and microphone on?
Yeah, I would just like to respond to some of that. And one thing to remember, Chris, and I think it was probably you that told me that there's a lot of technologies have come and gone, and CPR has stayed there robust and consistent throughout. And the thing to remember, it is the benchmark that everybody else is trying to emulate. And I think anything, and we had a bit of a discussion about this earlier today. I mean, anything is to augment, it's using that word augment, but and sort of build on what is already an excellent program. But I mean, I think you make a really important point as well, Chris, around the amount of funding that's put into sort of developing these new technologies could fund several CPR programs. But I think ultimately that's what funders want to see. Is where it's going in the future, and how we can actually get to a level where we can start forecasting the global ocean. And to do that, you're going to have to really find ways where you can get much more automated before you can get to a level where you can do that. There was and Julie and I have been in these discussions with one of the UN Decade programs we're involved with, that, if there was as much investment went into forecasting the weather as there was to forecasting biodiversity, we would be able to do anything we wanted. If you think the amount of funding that goes into meteorological forecasting and modeling, it's just extraordinary. And you can find out, you can forecast what the weather's going to be like in a month from now, almost. Maybe not to sort of hugely, well, certainly not in Devon anyway, they always get it wrong. But so yeah, I think I'm not too concerned about it, because actually anything that's done along these lines just really sort of highlights the value of the CPR. So yeah, but I'm kind of preaching to the converted here.
Thank you, Willie. And we've got a question from John here, who says it's a naive whole organism biologist question. A very good question, though. How much of an issue could the eDNA of extinct marine life be in the use of this technology to tackle biodiversity questions?
Yeah, so it is an issue. So especially in sediments, right? There's a whole field called ancient DNA that's looking at old DNA. So yeah, it's a problem, but in most pelagic realms, especially in the surface ocean where there's UV light, free DNA degrades pretty quickly. And I think that with bacterial degradation, they've shown that that eDNA has a lifetime of a few days. I think it's 48 to 72 hours usually, but that's just the typical eDNA sequence. So there's definitely going to be some there that's older.
Great, thank you, Julie. We've got a question here from Phil as well, who's just asking, the key is the human resource, as was mentioned earlier, and if you lose the interest of the next generation, you won't have taxonomists. And just from my perspective, as I mentioned about the Science Journal for Kids, it's engaging sort of different audiences with our science as much as possible. But I wondered if any of the panel have got anything they'd like to add to that.
I think that you guys have these gorgeous images, right? So you think of Richard Kirby and all these beautiful plankton images, and that is the stuff that makes people do marine biology. Nobody's looking at sequence data and saying, I want to be a marine biologist, right? So I think that there is that inspiration there. And what I don't understand is, I mean, for us, I think it's obvious, right? But I don't understand how that isn't, you know, it's always NASA and astronomy, and that's the cool, you know, looking at the stars is the cool thing to kids. But plankton, I mean, they're right here, and they're, you know, you need a microscope. But yeah, they're so much more interesting to me, and I think, yeah, their beauty is just, yeah, it's. So I do think I think it is there. It's just a matter of, yeah, the long-term training, I think, once they get to college and things like that, like keeping that interest.
Thank you, Julie. And is there anybody else that would like to add to that at all?
Yes, I just want to add something. And I mean, I agree with what Chris said about the technology and keeping the core of the CPR intact, because at the end of the day, this is the strength of the CPR. But technology can attract young people too. This is very important. We are talking about, for instance, taxonomists, but the taxonomist of yesterday, they might not be the same as the taxonomist as tomorrow. When you see the AutoCam, when you see the FlowCam, we need taxonomists to work on those data. We need taxonomists to help artificial intelligence to automatically identify taxon. And to me, the future of taxonomy, it's also having taxonomists connected to the molecular side of things, taxonomics connecting to the imaging side of things, taxonomics coding AI to identify things. So maybe it's because we have a quite an old vision of what a taxonomist is. I think taxonomists are there for a long time, and they will be here in 90 years for sure.
Great, thank you. And one of the taxonomists that we have got here has been around some time. That's Gerald Bolt. You've got your hand up. Do you want to ask a question there, Gerald? I don't know if you can unmute your microphone, Gerald. I'm not sure if it's working at the moment. David, did you have your hand up? And we'll come back to you, Gerald.
Yeah, it was just very quick, just to sort of agree with Pierre. You know, it's not just sort of the plankton work, but actually there's lots of areas of marine research, as well as probably terrestrial and all sorts, where people are using image analysis. And a lot of the work is really focusing on sort of producing, you know, databases of images, image libraries, and all of those all those images need to be checked by taxonomists and signed off. And that's not going to go anywhere soon, you know. So we're going to need taxonomists for a long time yet.
Great, lovely. I don't think we've got any more questions specific to this theme, but as we're sort of coming to the end of the day, does anybody have any sort of wider questions about the CPR, or any thoughts about the future of the CPR, or even any comments about today and the birthday? Then please feel free to raise your hand or put a question into the Q&A function. We've got some messages coming through. Just to note about entering into that survey. So if you fill in that survey, you also get entered into a competition to win one of Debbie Mason's prints as well, which were shared on the slide in between the themes. So that's there, and I can't see any more questions. Anybody got anything else they'd like to add? No? Well, that leaves us with all our questions and answers and discussions for today, which is wrapping us up with pretty much perfect timing. We've been on time for every single theme today. So thank you everyone for being incredible timekeepers. It's been a fantastic day. Thank you to all the people behind the scenes. I'm going to pass you back to David now for any final comments for our CPR90 celebration, and I hope to see you in 10 years' time.
Thanks, Maya. There's not much else to say from me, just to say thank you very much for everybody for turning up today and giving up your time to hear what we've been up to and how we wanted to celebrate our CPR 90th. Not ideal that we couldn't meet in person, but you know, I think we've made the best of it. Yeah, amazingly, it's all gone to time, and I think actually having people send their speeches in was that's definitely the future. You know, no timing issues there. That was great. So I think that's probably it. So unless anybody on the panel had anything else to say, I just echo what Maya said, and let's look forward to the 100th.
Thank you.
Thanks very much, everybody. We look forward to welcoming you to the MBA as soon as possible, and we'll enjoy the cake on your behalf. So thank you for joining us today.
See you all later.
Bye.
Bye.